Hey everyone, this is video number 8 for Module 7. This is the last video for this module. And in this module, I want to extend what I covered in the previous video. And mostly this is going to build upon the information about epigenetic modification and discuss how those modifications are involved in regulating gene expression. And I picked a number of phenomena that have been shown to use epigenetic modification just to illustrate what's going on and how that's involved in gene regulation. So let's take a look at the outline here. In black is what we discussed in the last video, and in red, part C is what we'll discuss in this video. Basically examples of how epigenetic modification is involved in gene regulation during development, how epigenetic modification produces differences in genetically identical individuals, for example, identical twins, epigenetic modification that occurs in response to environmental conditions, and I chose the example of a kind of a classic experiment showing nutritional differences in mice and how that produces epigenetic differences. And finally, a phenomenon we've talked about before earlier in the course, X-inactivation during female mammalian development, how one X chromosome gets turned off. And that's the mechanism by which that occurs is through epigenetic modification. All right, well, let's get right to it. And talk about the first topic, how epigenetic modification is involved in development. So first, some basics about development. During development, a fertilized egg, the zygote, divides extensively, and as development proceeds, cells become more restricted in their developmental potential. So the zygote obviously can conform all parts of the developing organism. And so that's referred to as being totipotent, having the potential to form the total of all organs, tissues, cell types. Okay. As the embryo develops, cells are less able to produce, they become more narrow, I guess I should say, in terms of the number of different cell types they can produce. So there's this progression. They become more restricted in their developmental potential. And also, as they develop, they become more specialized in terms of the types of proteins and the types of activities they're able to carry out. The end product of development for most, not all cells, but for most cells, is that they are what's called terminally differentiated. They are specialized cells, and they have one potential. They're unipotent, and that is they're a specialized cell that carries out a particular function, such as a muscle cell that contracts, or a neuron that conducts electrical impulses. Now, during differentiation, this narrowing process is going to turn off most genes in the genome. And only a few minor subset of all the total number of genes are going to remain on and needed for a specific type. So, for example, in a muscle cell, genes that are needed for making electrical signals in neurons are turned off. But genes that produce proteins involved in muscle structure, like actin, myosin proteins, those genes remain on. Okay? So during this process of development, is epigenetic modification involved in this process? Yes, it is. Epigenetics is involved in the process of turning most genes off. And this slide is just illustrating the process, how fertilization produces a zygote. The zygote is called totipotent, can form all structures in the adult, all cells. As it develops in the embryo, now there are more cells, but these cells are going to be more restricted in their development. Some of these are going to be able to form some structures, others are going to be able to form others. None of them are totipotent anymore. They're referred to as pluripotent. They can form a number of different structures, but not all of them. As development proceeds, cells become more restricted in their developmental potential. In the adult, you have some cells called stem cells that are present in different places. For example, in the bone marrow, cells that form blood are able to form all different types of blood cells, but not anything else. So they're called multipotent. And then cells that are restricted, terminally differentiated cells, not really shown on this slide, they're called unipotent. Okay? Does epigenetics play a role? So I already answered that question, yes, but how does it play a role? So we're going to look at the process for a gene that gets turned off, let's say. In a muscle cell, it's going to turn off all genes that are going to be necessary in neurons. What's going to happen if you look at the promoter of this gene, whose fate is to be turned off, and can we determine any epigenetic differences in the promoter of this gene from cells in early development, the zygote or early embryonic stage, versus adult? Okay? So, first of all, in a pluripotent cell, let's say this is an embryonic cell, versus a unipotent cell, this would be in the adult. Let's say this is the muscle cell, and this is a cell that was eventually going to form the muscle cell. If you look at the promoter for any gene that's fated to be turned off, and you sequence to determine the DNA sequence of that promoter, you'll find that they're identical. So the genetics is not different. Okay, there's no genetic difference going on here. If you look at the sequence, it's identical. That means it has identical sites for CpG methylation. And if you run through, they're all highlighted here. There's no difference in DNA sequence, and therefore no difference in the number and placement of CpGs between an embryonic pluripotent cell and an adult unipotent cell. Let's look at the pattern of DNA methylation, though. In an embryonic pluripotent cell, none of these are methylated, and so this promoter is not being repressed. And in an adult cell, however, all of these sites are methylated. So this promoter is going to be turned off, shut down, not accessible for binding to basal transcription factors. So this is a main mechanism by which genes are turned off during development as cells get a more narrow fate in terms of their developmental potential. They're going to be doing this to most genes that are not needed in that end fate. So for a muscle cell, all those genes that are not involved in an active muscle are going to be turned off, just like this case. Okay? So summary slide here is what's going to happen to most cells, most promoters in a cell going from early development to adult is they're going to, genes in those promoters in the genes, here are 4 genes shown along a chromosome, most of them are going to accumulate DNA methylation and they will be shut off. And so this methylation, we only, in the previous 3 slides, we only showed it for pluripotent versus unipotent, but this is going to become more and more progressive methylation until in the unipotent cell, this is an extensive degree of methylation. All right, so epigenetic changes through DNA methylation, anyway, is an important factor in development and turning most genes off during that process. How about in identical twins? Identical twins develop from a fertilization of a single egg, which splits into 2 in the first division, producing 2 separate embryos. So they are genetically identical. However, they're not phenotypically identical. If you know a set of identical twins, and you may be able to tell them apart if you know them well. And so what accounts for the phenotypic differences if they're genetically identical? Are there epigenetic differences between them? So there's a really good study that was published that looked at identical twins. And what this study did was to look at a pair of identical twins at 3 years old and map the epigenetic modifications along 4 different chromosomes in each individual. They looked at chromosome 1, chromosome 3, chromosome 12, and chromosome 17. And basically, they used antibodies to show, let's see, what they were looking at was DNA methylation. I think, right? DNA methylation? Yeah, DNA methylation along the length of the chromosome. Okay? And if the antibody stuck to a site on the chromosome, that means it was methylated. If it didn't stick, that means it was not methylated. So what this shows are the 2 chromosomes from each twin. Chromosome 1, twin 2. Chromosome 3, twin 1, twin 2. 12 and 17. And the staining pattern here, what it means, let me give you kind of the simple version. If it stains yellow, that means they're identical in staining pattern. Okay? If it stains red or green, that means there's a difference. Either one is methylated and the other's not, or vice versa. Okay? So, here's the situation for 3-year-old twins. You can see chromosome number 1 here is mostly yellow. There's a region where there's not much staining at all. I'm not sure what that means, to be honest with you. There are some limited sites in green and red, indicating some differences, but also another yellow region here. Chromosome 3, there's some green, but also some yellow. Chromosome 12, lots of yellow, very few spots of green and red. Excuse me. And chromosome 17, almost completely yellow. Let's look at the situation 47 years later, 50-year-old identical twins. What you can see is there's virtually no yellow here, so mostly green and red for all 4 chromosomes. And that indicates that there's extensive differences in epigenetic modification of these chromosomes by DNA methylation. And so the conclusion from this experiment is that even though they're genetically identical, the epigenetic modifications are vastly different between identical twins. And so they don't know exactly why that is, but they suspect some kind of environmental difference between twins can cause these epigenetic differences as they age. Here's another example of identical twins. Scott and Mark Kelly. Scott Kelly went into space on the space station while Mark Kelly stayed on Earth. And what they did is they took samples, blood samples, and analyzed their DNA and blood cells for a couple things. What is the length of the telomeres? And what are the epigenetic modifications that they could find for specific sites? So they did this, took these samples beforehand, and then they took them again right as Scott returned back to Earth. Here are the main results. They found that Scott, who was the space twin, his telomeres lengthened while he was in space. So it's like he was not really aging, because as you age, your telomeres shorten. And they looked at this in somatic cells, by the way. But upon returning to Earth, his telomeres returned back to the pre-space length. Okay, so that's kind of a curious feature. I don't know why that's true, what happened exactly, but that was the observation. For the epigenetic modifications, they found a similar situation. While Scott was in space, epigenetic modifications cropped up that were not seen before he left. And then when he returned, many of those went away again, and they returned back to the Earth status. Not all of them did so, however, so some of them persisted. So the conclusion for that is there's something about Scott's experience in space that that produced these changes relative to Mark's epigenetic changes on Earth. What could these be? Any kind of environmental change you could think of being in space. Difference in gravity, zero gravity. Change in nutrition, maybe change in stress. The exact cause is not known, but there is some environmental difference that's producing that change. All right, so related to that, while we're on the topic of epigenetics and environment, let's talk about examples of how environment is thought to produce epigenetic modifications. So there are all kinds of studies in the literature now about how nutrition, like nutrition itself, let's say a very nutritious diet versus a junk food diet, is that going to produce a difference in epigenetics? Or chemical exposure, will that produce a difference in epigenetic modification? Or physical factors such as temperature, or psychological factors such as stress or trauma. So there are the huge number, kind of an explosion in this field of environmental epigenetics. So I chose one example kind of a classical experiment which was done in mice. And this looked at changing the diet in the mouse to see, excuse me, to see if there are any epigenetic changes that occurred as a result. So here's the setup of the experiment. This experiment used yellow mice, and we've talked about yellow mice before. If you remember, yellow mice is an allele of the agouti gene, and yellow mice are variable in their phenotype. They're not uniformly yellow. They go from pretty much homogeneous yellow all the way to almost agouti, which is brown, and everything in between. So slightly mottled, and heavily mottled. Okay? So we would say that the agouti phenotype shows variable expressivity. What's the reason for that? All right, so this is where the experiment comes in. They used agouti yellow mice and fed them different diets. One was a control diet, which is the standard mouse chow, I guess you would say. And that's what are normally given to lab mice. Okay. The other, another group, an experimental group, got a set of a mouse chow supplemented with substances, chemicals that are considered to be methylation precursor molecules. This includes folic acid, vitamin B12, and choline, basically methyl donors. Okay. And the other experimental group was fed toxins, various types of toxins, one of which was what are called endocrine disruptors. These have been found in various herbicides that have been sprayed on crops and shown to cause illness in farm workers that are exposed to them. Okay, what were the results of this experiment? Let me just show it on this slide. So what was fed actually was a yellow mouse female who was pregnant. Okay, so when she was given normal mouse chow, these are the variable offspring mice that she produced. Okay, so that's the typical observation for yellow agouti mice. They produce variable phenotype from homogeneous yellow to agouti and in between mottled. And when they looked at the promoter for the agouti gene, what they found was that the promoter, which has a CpG island and can be methylated in yellow mice, offspring from the mother that was fed normal mouse chow, the yellow mice, the promoter was not methylated. So all of these open circles here are CpG sites that are not methylated. And so that means the agouti gene was expressed at high levels because this promoter was accessible for the basal transcription factors to bind and be transcribed. And the mice that were pretty much agouti brown colored, if they look at the promoter of this gene, they find that it's highly methylated at all CPG sites. And so it's shut off because it's not accessible to the basal transcription factors. If they looked at the mottled mice, they saw something in between, a little bit of CPG methylation. Not all sites are methylated, and a low level of expression. So there's a correlation in the mice of the offspring of the mother given the normal mouse chow. Low level of promoter methylation produces high expression and yellow color. Intermediate methylation, intermediate expression, and mottled. High methylation produces low expression and the agouti color. How about the experimental conditions? So in the mice that were fed, let's look at this one first actually, that were fed the methyl donors, choline for example, they found that the agouti promoter was highly methylated. They proposed that having these methyl donors in the diet allowed for higher activity of the methyltransferase and therefore higher methylation of the promoter. Low expression of the gene, and therefore most of the mice offspring were agouti in phenotype. They didn't see many mottled, and they saw no yellow mice. How about the mice fed the toxins, the endocrine disruptors? They found that there was not much methylation. So there was high expression from this gene. And most mice were almost all yellow, maybe slightly mottled. So the conclusion from this experiment is that nutritional conditions can have an effect on the epigenetic modification of a promoter such that it can push the promoter to be not methylated and highly expressed, or highly methylated and not expressed. This is not due to a difference in the gene sequence itself, not due to genetics, but entirely due to epigenetic changes. So, nutrition has an effect on epigenetic modification, and therefore gene expression. Okay, how about the last example? How does epigenetics play a role in the process of X-inactivation? So if you recall from way back in the semester, we talked about how in mammalian development, one X chromosome early in development gets inactivated, and this occurs in every cell in a mammalian female, such that each cell has one active X chromosome and one inactivated heterochromatic bar body. Okay? And the inactive X, it turns out, is inactivated into heterochromatin by epigenetic modification. And we can see that in an inactivated X, this bar body here, because it's staining with antibodies against epigenetic markers that occur during inactivation of a chromosome. How does that happen? The mechanism of X inactivation involves each X chromosome has a region called the X inactivation center, XIC, part of the long arm of each X chromosome. So this is the XIC, and there's a gene within the XIC called the X inactivation specific transcript, XIST. What is the XIST and what does it do? Characteristics of the XIST are, number one, it's located in the XIC. This gene is transcribed only from the inactivated X chromosome, not the active X chromosome. This gene is transcribed into 20 kb long transcript, very long transcript. However, this has no protein coding region. So it's transcribed, but not translated. The RNA that's transcribed does not leave the nucleus. It doesn't need to because it's not translatable. And what it does actually is it coats the surface of the X chromosome from which it was produced. this coating on the surface is going to eventually result in the inactivated X chromosome that produced it being heterochromatized and X inactivated. So it works something like this. From a chromosome that's going to be inactivated, it produces XIST. The other chromosome does not. XIST is going to coat the surface of this chromosome. And this coating somehow causes heterochromatin to form on this chromosome. The exact mechanism is not known, but what's thought is that this mRNA, the XIST RNA, is going to attract enzymes that are going to methylate the DNA and are going to methylate the histone tails. And so that's going to form, allow the chromosome to form heterochromatin to pack very tightly and inactivate it. Now, this slide is just showing if you look at a female cell after X inactivation has occurred and you stain it with an antibody that's specific for unacetylated histone H4. So unacetylated would be present in the heterochromatin. You will find that the inactivated chromosome is staining with this stain. It stains red, whereas the active X chromosome is not. So the inactivated X has unacetylated histone, and that correlates with inactivation. This slide shows that DNA methylation also occurs during X inactivation. This is before X inactivation, looking at DNA methylation in promoter regions of 4 genes along the 2 different X chromosomes. After inactivation, the inactivated chromosome with a little I superscript here shows DNA methylation in the promoters of these 4 genes, whereas the active X does not show DNA methylation. So during X inactivation, there's both DNA methylation and deacetylation of histones, and that's going to contribute to heterochromatin formation on the inactivated X. All right, and just a couple slides to summarize here. Epigenetics is now a very active area of research, and this is tying into gene expression and other things as well. And so we talked about how nutrition can affect gene expression through epigenetics, the mouse example, but environmental effects also can play a role, and people are experimenting with how environmental pollutants or toxins can have effects and what specific effects they have in terms of epigenetic modification and gene expression. Also, behavioral genetics and exposure to trauma. How does that affect epigenetic modification? And even in certain psychological disorders, there have been ties to epigenetics. And here are some recent papers investigating epigenetics. Other epigenetic tags related to sexual preferences, homosexuality versus heterosexuality. That's an area of active research. And how about substance abuse? Does substance use or abuse result in any kind of epigenetic modification? So this is a very up-and-coming area of research, and I'm sure will have extensive influence on the field of gene regulation in the future. Well, this ends Module 7, so I'm going to end this video here. See ya.